Okay, it is 9:35, and I am going to take a moment to introduce you. So thank you everyone for joining us. I'm hosting the Center for Genes, Environment, and Health seminar series. This is a seminar series where we're interested in the overlap between precision medicine and disparities and populations underrepresented in medicine and medical research. And today, we're really happy to host David Conti, who's a professor at USC. And David's been at the forefront of this issue, both from a methodological perspective and also from a kind of applied perspective in the area of prostate cancer, where, as many of you are aware, there are tremendous disparities both in the incidence of prostate cancer and in the outcomes of prostate cancer, particularly higher rates of higher incidence and higher mortality in African-American men. So without further ado, I'm going to let David take over and tell us about his work.
Okay. Thank you, and thank you for inviting me. Oops, 1 second here. Let me make sure I share the proper screen here. Okay, hopefully you should be seeing the title screen in full there. And so, okay, I'm going to talk today about a Trans-Ancestry polygenic risk score, and really just sort of highlighting sort of where we were a few years ago with the genetics of prostate cancer, what we've done just recently in terms of a lot of sort of extending that polygenic risk score so it can be effective across multiple ancestry groups, and then how it can be improved and modified. And so some of this will focus on the methods behind it and what we use to get there. And then others will, we'll talk about sort of the practical aspects of applying this in a real world situation. So as Eli pointed out, there are differences by race and ethnicity for prostate cancer incidence. And on the left panel here, you can see that this is the incidence rate ratio, and this line up here that's hovering around 1.6 or so across the last 20 or so years is the incidence rate ratio of individuals that self-identified as Black versus white as the reference here. And you'll also notice that on the other side, around 0.5 for the rate ratio is Asian and Pacific Islander groups. And so there are differences in the incidence of prostate cancer across different race ethnicity groups. Of interest is the actual incidence rates have changed over time, but the incidence rate ratio is remaining pretty constant. In terms of, there's also mortality differences, and those are a little bit more extremes in terms of individuals identified as Black versus white for the SEER data, and how that compares across time, and there's still sort of substantial differences. And so that it's really this information that motivates a lot of what we've been doing. And if we take a step back, just so we're all on the same page of what are the goals of genetic association studies, and the lofty goal, I think, would be really to identify the causal variants for a given disease, right? And that's really what maybe we're aiming to try to do. And that, maybe has several steps to that process. Discover regions of the genome that harbor risk loci. Can we rank SNPs as putative causal risk variants within that with fine mapping type of techniques? Can we characterize the impact of these risk loci? What's the impact in terms of familial relative risk or heritability? On the whole, what's the impact of these? What's the biological impact? Can we, interpret these, go into the lab with those that we think are putative causal variants and figure out the biology and potential impact there. And then finally, the translation and the prediction. Can we use the combination of all the variants that we've identified as a genetic risk score for screening in the population, and then also for individual risk prediction? And this is sort of the Manhattan plot from the GWAS from a few years ago with Fred Schumacher, 2018. Just to give you an idea, there are about 20 or so loci in this figure that have p-values much greater than 10 to the minus 30. The blue here are those that were previously known, and the purple are these newly discovered ones with this particular GWAS. But we're getting up to, with this particular GWAS, of over 100 different loci or regions that are of interest. And so in constructing a genetic risk score, how do we do that? And this is where simply it's a weighted sum of the risk alleles carried by each participant, where the GRS is looking over the sum of the different variants. The G here is the count of the risk alleles, so whether it's 0, 1, or 2 of that risk allele, and then there's a weight associated. And typically this is the log odds ratio from some estimate, from some study. And this is a simple formula, but it really captures everything about what we're trying to do here is, well, what are the SNPs that we include in this sort of summation? What's the weight? How do we say that one SNP is sort of preferentially more important than another one in this? And then the real question here is, how does that change across populations? And, again, if we go back to, if we identify causal variants and they are present in different populations and individuals, then how do we weight those? They should be the same? Do we include them? But if we don't have that causal risk variant, is it LD that we're capturing with just an individual SNP? Can we use combination of SNPs to capture that? And so those are the questions of constructing, really, I think, in methods around sort of constructing genetic risk scores nowadays. Typically what we do is we're dealing with case controlled data. We're really interested in sort of identifying sort of in this case, the 25 to 75% average or normal range of this PRS here, and then comparing that to sort of the upper 10% or some extreme level there between cases and controls to evaluate this genetic risk score. So again, in doing all of this whole process, how does discovery, characterization, and translation differ across populations, and what are some of the aspects that really could influence things? And certainly there's differential linkage disequilibrium that may sort of alter what the weight should be or the SNPs that we would identify. Different allele frequencies, whether it's common or population specific, and how does that impact the sort of the ability to characterize population specific effects or across populations of different ancestries. Different allelic risk. If there is gene environment interaction, that would impact sort of how we assess what that weight should be or what the overall contribution of that particular variant is in that population. And outside social risk factors, environmental risk factors, what's the population delineator that gets captured, and how does that then modify this risk? Practical aspects of just performing GWAS in different populations. What's the differential coverage between population due to the genotyping arrays? In prostate cancer, we've been performing studies over the last 10 years. Arrays have developed over time. Sample sizes are getting increasing. Every population and every study has done different arrays. How do we incorporate all of that? And then, of course, different sample sizes. And then different variation in the trait. So if we're going to quantify something in terms of heritability, what does that mean when the overall trait variation might differ across populations? So going back to sort of where we were a few years ago in prostate cancer and with the ELLIPSE sort of consortium for this, the previous GWAS up to that point had identified about 100 known variants. And then we performed a GWAS with 79,000 cases, 61,000 controls. All of the individuals in these studies were individuals of European ancestry, and we were able to identify 62 novel variants. All of those, the 62 plus the 100, captured about 28.4% of the familial relative risk, assuming the familial relative risk is about 2 in this case. And then the genetic risk score here identified over 10% of the population with an odds ratio of 2. And we can see that here in terms of looking at sort of the risk categories of the polygenic risk score, again, compared to sort of this 25 to 75th percent as the reference. With using the known 100, we could get up to about 3%, or sorry, threefold increase with the 90 to 99 percentile, and almost a six-fold increase for the upper 1% with the known. We went through all of this with additional cases in individuals of European ancestry, and we were able to really just keep that impact of the genetic risk score here about the same. Really no change between adding these 62 new novel variants, even though they increased the familial relative risk, but they didn't impact sort of the performance of the polygenic risk score to a substantial degree within this population. At the same time, we were performing sort of ethnic specific GWAS discoveries, right? So within our African American sample, individuals of African ancestry, we had about 10,000 cases and 10,000 controls. And then we looked at just 100 reported known risk loci, and we were able to sort of categorized that, what percent were polymorphic. 94 of those were polymorphic in the African ancestry population. 81 of those were directionally consistent, and 47 are nominally statistically significant. And this really sort of just these little bullet points kind of summarize some of the difficulties is, with individuals of European ancestry, we had sample sizes up, 80, 90,000 in terms of cases and controls, and now we're down to 10,000. And performing an ethnic specific GWAS, we're really limited in terms of the power that we have. And so even within this context, we were able to identify 2 novel signals. They were in known regions, but they were sort of African ancestry specific alleles that we identified in 2 of the known regions, one of them AQ24. And so the impact of that on sort of a comparison here between European and African, if we look at sort of where we were with those 100 and then plus these 2 African specific, even then we were able to have sort of a polygenic risk score, a genetic risk score that worked pretty well within African ancestry individuals, with the 90 to 99 percentile having a 3-fold increase in risk and being pretty consistent across ethnic groups here. Another thing that we were doing at the same time was performing fine mapping, right? And if you go into any region, you see patterns like this, and I'm sure everybody on this call is familiar with these types of LD plots. And what we were using in all of these previous examples was really just sort of taking the SNP with the most significant p-value within each region and using that to capture that risk. But what we really want to do is go into each one of these and say, well, are there multiple independent signals? And if so, what are the best SNPs that capture that with fine mapping? And so we did that within the Practical ELLIPSE Consortium. There's a total of 54 studies, again, similar types of case control numbers in terms of 80,000 cases, 61,000 controls. We had individual level data on some of those cases and controls, but not for all studies. And so we needed a way to perform an analysis that would use the summary statistics so we could leverage the power of the entire sample size. And then within those regions, due to imputation and the arrays, we had about 70 million SNPs that were available. So we applied an approach that we developed called JAM, joint analysis of marginal summary statistics, to fine map these 84 previously known regions. So just 84 of them, because some of the regions like AQ24 are a little bit complicated to sort of sort out with these types of approaches. But within those 84 regions, we were able to identify 99 independent signals. So some of the regions had multiple signals, 12 regions, and some of the regions had up to 5 signals. And then we were able to also define sort of 95% credible sets. What are the sets of SNPs that are putative, functional, or causal variants within each one of those regions? The impact of those, when we sort of combine the new 99 independent signals with the others, is we increased the familial relative risk explained from 23.2% to 26% within this data set. And then adding the 99 independent signals raised that up to 30% of the familial relative risk explained. So fine mapping does have an impact in sort of capturing sort of what's going on within each region. And just to take a step back of sort of what did we do? This is just the joint analysis of marginal SNPs, summary statistics. And this is just a linear model to motivate this. And with a linear model, you can use least squares to estimate the effects. And that's all that's just here. And then all summary statistics approaches use this basic math. And what they're doing is really just substituting in for this G prime G here, a reference data set to get the correlation structure. And so that's very important that that reference data represents the data, the same individuals that you performed your analysis on in terms of the correlation structure. And of course, that's an issue if you're doing this across multiple populations and studies. The other part of this equation for least squares is this G prime Y, and that's really the summary statistics for the effect estimates. It's sort of an average sort of trait effect for each allele. And so you can get that from summary statistics, whether it's a z-score or the actual log odds ratio. And so this is the basic analysis that we do with summary statistics. Now, what we've done with JAM, and again, I don't want to get into the math too much, but I do want to highlight some of the things that of why we've done some of the things we've done is because the complication of highly correlated SNPs, we use what's called a G prior. And what that does is it uses the correlation structure in the prior itself. And so the impact of that on the analysis is that it inversely sort of downweights models where two SNPs are highly correlated together. And so it really helps us to sort of identify, well, what are the independent impact of these different SNPs? And we do this within a Bayesian framework for a stochastic selection where we're really just sort of flipping a coin of saying, well, what's the prior probability of including each one of these SNPs, and then we'll look at the posterior inclusion probabilities to identify sets of SNPs and which ones are the most likely to be the index SNP and which ones might be included in the credible set. So what do we get at the end of this is focusing on one of these regions from that meta-analysis at that time. These are the one at a time or the marginal p-values in the LD plot that you get from that. And there's 438 variants within this region that are genome-wide significant. And then when we run JAM, we end up with clear evidence for one signal, and it's one SNP that captures that signal. So this is sort of the best case scenario that you would have in sort of performing sort of fine mapping here as you go forward. And then, but much more common is sort of a peak where you have, this peak looks very good in terms of sort of being narrow and pointing to a certain region. There's 444 variants here that are genome-wide significant. And when we perform JAM for fine mapping, there's evidence that points to 4 independent signals within this region, but they're split across about 10 or 11 different SNPs. So each one of those could be used sort of in constructing a genetic risk score. And so which one do we use and why do we use which one, right? And how do we then apply those across populations? Those are some of the issues that we need to think about. And so additional information will be crucial in sort of picking among those 10 which ones to include. And that additional information can be functional or it can be from other populations. And so that's where we really were thinking we got to combine all of the data that we have out there. And I'm sure people have seen these figures that just show the lack of diversity in GWAS studies. And that's going to contribute to health disparities if we want to move forward making polygenic risk scores for population and individual screening. And you can see that, that the majority of the studies over time have been in individuals of European ancestry. And we really need to just increase this non-European ancestry to do that. And it just as the result of that, in looking sort of the polygenic prediction accuracy relative to individuals of European ancestry, you can see all of these in every other population for at least this example of 17 quantitative traits, they're all below 1. So in all populations, it doesn't reach sort of the ability for prediction in your individuals of European ancestry. And that's really just unacceptable. And it's just driven by the fact that we're doing discovery in individuals of European ancestry. And so we performed a Trans-Ancestry GWAS meta-analysis for prostate cancer, and this combines the data across 4 different sort of ancestry groups: African, East Asian, European, and Hispanic. And you can see that it's 177,000 individuals of European ancestry and only 21 or 27,000 individuals of African or East Asian, respectively, and very few individuals from Hispanic populations. But with this data, we were able to perform sort of a ancestry-specific GWAS, and then we performed a meta-analysis across those 4 different GWAS. And we were able to discover now an additional 86 novel risk variants, 62 of the known regions. We were able to identify a stronger marker for using fine mapping. And then 121 of the known variants remained sort of the lead signal in each region. This also includes ethnic specific variants. So we didn't just take the top from the meta-analysis, we looked at each individual sort of ethnic specific GWAS and incorporated variants there. And we did this through an approach using JAM that really combined the conditional estimates across the multiple populations, the 4 different population summary statistics. So we would go into each, we would start with a SNP with the meta-analysis across the multi-ethnic meta-analysis, and then we would add a SNP doing forward selection using JAM. And then we would go back to each population and ask what's the conditional estimate and how we would combine them. And this process took about a year. We would go into each region, look at the LD plots and ask, well, what's the conditional statistics? What are we looking at? What's the SNPs that we need to include? Is there a SNP that sort of isn't represented in one population that we need to include, and so on and so forth. And one of the biggest practical problems in doing this is that in some of the populations, because of coverage or because of the just allele frequency differences across populations, is SNPs don't exist in our data set from one population to the next. As an example of that, including the East Asian summary statistics, we lost almost 30% of the SNPs that we were trying to do an analysis on. And so it really becomes a sort of a painstaking way of going through this. And I'll come back to sort of ways that we think we can improve on that. And so,
David?
yes. Yeah.
So just a quick question, though. I don't know if you maybe you'll get to this later, but when you said there are population-specific variants, are these generally kind of things that are not that just are polymorphic in only one population?
Yes.
Or do you have a lot of examples where the variant exists in all populations, but it's only a risk factor in one population, and it's not like a linkage disequilibrium problem that you're just choosing the wrong variant?
No, great question. And we have examples of all of those. We have examples where it truly is a population specific allele. It only exists as polymorphic in one population versus others. Then we have some examples where, yes, it exists in all populations, but the effect is only prominent or sort of non-zero in one population, right? And that's actually a great segue into why we constructed what we call a multi-ethnic GRS, and we used a weight that was from the multi-ancestry conditional estimates, so those estimates, and instead of doing a inverse variance weight of all the populations, we used a z-score weight. And the reason for that is because some SNPs had effects in one population and not in others. And if we used a fixed effects meta-analysis, it would downweight the weight of that SNP. And we wanted to keep that one population representing sort of that effect or that impact of that SNP. And so that's why we use this z-score weighted meta-analysis for this. To sort of capture that. So it was 269 variants with a multi-ethnic ancestry weight. And we're using the polygenic risk score. So it's, for any individual, it's the same risk score. I know some groups are sort of developing maybe population-specific GRS. This is sort of one GRS. And it's just if an individual comes in, that allele is present, then we calculate that GRS that way.
I have a question. This is
yes.
Catherine. Sorry. So it was very interesting, the JAM result. So is that I have the same assumption of a single causal variant during fine mapping, or do you have an assumption of multiple causal variant in the region? No,
so we specifically are looking for multiple causal variants within the region. So we really do just a forward stepwise selection of sort of combining different SNPs within a model. And then, some of the regions we had up to 5 SNPs in a model, and then we're using the conditional estimates across the multiple populations to get that.
Is the conditional estimate also built into JAM, or you just have to do iteration by yourself?
No, we did it at that time. We did it as fitting JAM within each population. and then meta-analyzing the conditional estimates, and then going on to the next step of a forward selection and building it that way. I will come back to that because that's great. And we now have a way to do that, sort of what we call multi-ethnic JAM, that naturally incorporates that. So you don't have to worry about the independent sort of modeling problems.
So David, I have a quick question. This is John. So does this mean that if someone gets the summary statistics from this paper, it's kind of hard to figure out these? I mean, like, you just need to, if you want to do the weight, you just take the weights from the paper, and because you're not going to recreate them.
Yeah. I mean, that's the unfortunate thing, is the way that we constructed this, by going by hand and looking within each region, it is not very reproducible, right? And the unfortunate part of it, I think we did a very good job by spending the time of looking at this, you know, those who've worked with Chris Hyman, you know, I mean, we spent time looking at every little LD plot and across the populations to do that. So I think we did a good job, but I think it makes it hard for saying, well, let's reproduce this. That said, I think we are getting there with sort of the modifications we've made with multi-ethnic JAM and how to implement that. And that comes very close to what we get by what we were sort of duct taping together previously. Yeah, great question. Okay, so how does this GRS look within each population? And so if we focus just up here on the upper right figure, this is within individuals of European ancestry. It's the density plots of controls and dotted line cases in the solid line. The x-axis there is actually just the PRS value, but because it's a weighted log relative risk sum, we can convert that to a relative risk value just for clarity on the x-axis labeling. And so if we look at sort of a cutoff here of where a twofold increase in risk of cases would be on the PRS scale, what that does is it captures about 51% of the cases and 20% of the controls. So that's that relative risk we were showing sort of in the categories, but this is just sort of saying, well, at a twofold increase, how many cases versus how many controls do we capture? If we look at that across every sort of ethnic group here, it's sort of, they're all standardized to the control distribution within a population. And then we look at what the twofold increase is for that population. And so for African ancestry, we capture 45% of the cases and 20% of the controls, okay, at that level of risk, sort of. And then for East Asian, it's 46% of the cases, 20% of the controls, and then Hispanic, 47%. So we are capturing a very similar amount of sort of percent of cases across the different populations, sort of, if you were sort of to put that down at a 20% control level. So within each population, we do get similar distributions and similar ability. So, you know, the other way to look at this is, again, as we've done previously, is to look at sort of the PRS or GRS categories, comparing now to the 40th to 60th percentile. And now we have a little bit more data, so we've done it in terms of deciles. And in the top 90 to 100 percentile, the estimates across all populations hover around 4. And now this is in the data, the individual level data that we had used in the multi-ethnic GWAS. So there is the potential for a little bit of a sort of winner's curse, letting these estimates be a little bit high, but we do see consistently in the 80 to 90 around 2.5, and then the 90 to 100th around 4. Looking in additional populations that were not included in the multi-ethnic meta-analysis, so this is Partners and MVP and the UK Biobank. When we look at those, we see very consistent estimates for the individuals of European ancestry, all around 4, about 2 to 2.5, ranging from those. And then individuals of African ancestry, we have several studies. Again, notice the sample sizes, although MVP is getting up there in numbers, but we do see a range now in terms of the replication and the ability of this PRS to capture sort of the 90 to 100 percentile. Now we're ranging in a 3 to 4-fold increase. But there's something else going on behind here, and that's really the age distribution of each one of those studies. And what we've seen is that the polygenic risk score is really modified and has an interaction with age. And so it's really only for the upper 10 percentile and less than 55. So individuals less than 55 in terms of cases, the 90 to 100 percentile, the risk estimate is up to 6 and almost 7 fold increase compared to 4 for if a case is diagnosed after 55. And we see sort of the same trend looking in individuals of African ancestry as well. So then if you look marginally and you don't account for age, you actually then the risk estimate will fluctuate based upon the age distribution of your cases in the studies that you have. And certainly that's a big issue if you don't have, in some of the studies that we have of African ancestry, we don't have a large sample size. And so the age categories due to sort of the sampling process can be different. If we look at aggressive versus non-aggressive, the genetic risk score does not differentiate again between aggressive and non-aggressive. So we've done very well for identifying who is going to get prostate cancer, but we cannot identify who gets aggressive prostate cancer with this polygenic risk score. The estimates within non-aggressive to aggressive are the same if we compare the controls. And if you make a comparison between aggressive and non-aggressive, there is no risk difference between those in the polygenic risk score for the common variants that we have in our polygenic risk score. Now, if we compare polygenic risk distributions or genetic risk distributions across populations, what do things look like? And here it's a similar type of figure, but what we've done is we've standardized sort of here to the European control distribution. And then we've looked at what the difference is in the shift of the polygenic risk difference for the different ancestries. So here we see that 20% cutoff for Europeans actually has 54% of African ancestry individuals would be identified above that point. Okay, and we look at the sort of East Asian ancestry, same sort of cutoff, 20% of Europeans being above that polygenic risk score cutoff would only capture 9% of East Asians. Okay, and then Hispanics, the distributions are very similar. So what this suggests is that, well, we really need to take into account the ancestry, okay, to get a relative reference panel sort of the polygenic risk score to identify somebody's individual risk, right? So this works well within populations, but how does it compare across populations in terms of sort of absolute risk? And then that's really sort of really summarized nicely here is what we've done is we went back to the individual studies that we included in the multi-ethnic meta-analysis, as well as supplementing with the 1,000 genomes populations. And we calculated the mean of the PRS. Here's on the sort of the absolute PRS scale on the y-axis, and there's the different populations ordered in terms of East Asian, European, Amerindian or Hispanic populations, and then African ancestry populations here on the right. And then the solid dots here are controls or population distributions of the means. And then the open dots here are cases from those same populations. And you can see that the mean difference differs across populations, but also is substantial between sort of cases and controls across those within a population. And so if an individual comes in and has a score of, let's say, 25 on the absolute polygenic risk score, genetic risk score, how would we characterize their risk, right? It has to be ancestry specific in how we would characterize that individual's risk. But again, once we go to ancestry specific, it becomes a relative risk. The difficulty is in capturing the absolute risk of that individual. And how do we define that absolute risk? And I'll come back to that here in a second. But another way to look at this in terms of sort of how the genetic risk score might be used in screening is to go forward and say, well, at a top 10% of the polygenic risk score, the genetic risk score, we would capture 30% of individuals in European ancestry of the prostate cancer cases, and it's 30% of the lethal cases for that. And then on the flip side, this is if we looked at the bottom 10%, we would capture 2% of those prostate cancers, because with prostate cancer, it's also important to identify who doesn't have sort of a high probability of having prostate cancer. If we look at African ancestry individuals, we can see the top 10% is about 27% of the prostate cancer cases that we would identify. Now, to put this in a different scale of, well, what is that lifetime risk? And here we have to have some sort of outside information, reference information that we pull in. And that's this, in a European ancestry population, overall, there's a 1 in 8 chance of a man having prostate cancer in their lifetime. Now, if we sort of stratify that by sort of the PRS risk, what it says is that the top 10% of the PRS risk is a 1 in 2.7. Okay, men will have prostate cancer. On the flip side, the bottom 10% is 1 in 40. Okay, and all of that is just sort of taking the risk estimates that we have from the multi-ethnic sort of GRS, and then stratifying and spreading out that European ancestry of sort of 1 in 8. And we do the same sort of with the African ancestry. Well, that's 1 in 6 is sort of the baseline in the population. And let's spread that out then by the polygenic risk score. And we get a 1 in 2.2 men for the top 10%. And on the flip side, it's 1 in 20 for the bottom. So we've taken the genetic risk score, now we put it into sort of a lifetime risk. Another way to do that is really looking at absolute risk by prostate cancer. And here we've done the same thing, and that's represented, if we focus on sort of the left panel here within individuals of European ancestry. This black line is the absolute risk in the overall population. And we get that information in this particular case from SuSiE, using mortality and incidence rates. And then again, we apply our risk estimates for the different categories, and that just sort of spreads out that line proportionally. And again, we use proportional hazards as this goes forward in time, and we're able to develop these sort of cumulative absolute risk curves across the age for individuals. And you can see that for individuals in the top 99 to 100, they get a lifetime risk of 65. At the age 85. And we can do this for different populations. But what we're doing is we're going to the SuSiE database and we're saying, hey, give us the information of mortality and incidence by self-identified ethnicity. And now we're going to use risk estimates that we've identified within our sort of African ancestry, European ancestry, Asian ancestry, and we'll stratify that out. And so in combining those, are we really capturing that risk difference in a population? And so even though I've been arguing that our genetic risk score does well across ancestry groups, if we combine it with the absolute risk from SuSiE for African Americans, that absolute risk does have an increase, okay, relative to Europeans. But when we put it with the risk estimates we get from the genetic risk score, we end up with the same lifetime risk. Okay, we do see sort of less for Asians, about the same for Hispanics. And so what we're trying to capture here is what happens when we take an absolute risk, a genetic risk comparison within a population and put it on an absolute risk scale. So moving forward, how can we improve the genetic risk score? And one of the things we looked at was, well, can we do a genome-wide risk score? And quickly here, what we did is we ranked the SNPs that we identified in our multi-ethnic meta-analysis by p-value. And we asked the question, well, if we take the top SNP and we include that, how well does that top SNP perform? And now let's take the next two and put those in a genetic risk score, and so on as we go down the list here. And the first 269 are our genetic risk, sort of polygenic risk score that we've been talking about. And as we include each one of those SNPs, you can see that here, this is the estimate of the odds ratio in the 90 to 100 percentile in European individuals, and that goes up. And then it ends up being the estimate that's around 4.2 or so here in Europeans. We also have an AUC curve that goes up there over a little bit over 0.83 with the 269. So then as we go down this list and we start including independent SNPs by lower and lower p-values, how does that change the performance? And it really doesn't. It doesn't really improve sort of the AUC as we go forward. And it actually, as you start to include more and more SNPs, the noise takes over and it actually hinders the performance. And again, this is using our multi-ethnic p-values, but going into an independent data set, the UK Biobank, to evaluate its performance. And we've done a similar thing within African ancestry individuals, going into a Uganda study that we have data, 1,500 cases, 1,000 controls. Same sort of thing. There's much more variability. Again, smaller sample size. Again, probably representative of discovery within European individuals, European ancestry individuals, but we have sort of a lot more variability. But that variability decreases a lot in sort of the performance as we include more and more noise. Okay, and so 1 second. So I mean, I think the conclusion here is that maybe GRS, genome-wide will not improve prediction, but this is using a very simple approach. There are other approaches, and we've been looking at sort of LB score and some of these other empirical Bayes approaches to do genome-wide, and we're not seeing an improvement, and we actually see poorer performance than the 269. So did somebody have a question?
Yeah, I had a quick question, actually related. So when you get up to the first 269 vertical line there, that's using JAM all the way.
That's using JAM.
And then at that point, you start to rank by p-value. Okay.
It would be, I mean, you could imagine doing conditional analysis genome-wide and then getting that might.
Well, that's kind of where I'm going, because when you start looking at, I'm wondering whether the work showing that LD-pred or the Bayesian approaches don't improve, whether it's a matter of kind of like there's some kind of JAM / Bayesian genome-wide type approach that would take the best of both.
Hold that thought. I think that's a wonderful idea. So we'll get there. So other ways that you can improve this is really just increasing the sample size, right? And that's what we're doing right now, sort of in process. And we saw that from going from what the Schumacher paper in 2018 to where we are now is the 2021 improvement in all of the sort of odds ratios for the 90 to 100 percentile, but the improvement was sort of larger in the non-European populations, and that's because we added non-European populations to Discovery. And so can we do that again? And that's what we're sort of undertaking. So this is where we were with the Discovery in the multi-ethnic that I just presented, and now we have increased the African ancestry individuals to 19,000 cases and 63,000 controls by including UK Biobank, MVP, all of these studies here. We've increased sort of the Asian case numbers slightly, and we're doing the best that we can to increase other populations. We've just finished sort of this Trans-Ancestry meta-analysis, and we have 100 new novel risk variants, over 100. And so we're doing conditional analyses and sort of constructing a new polygenic risk score to evaluate that. The other thing, and this gets back to sort of what we've talked about earlier, is multi-ethnic fine mapping. And, again, how can we sort of include multiple ethnic groups and this other information, and really the big advantage here is this differential LD, and really hovering and leveraging that so we can identify sort of common variants that are found across populations that then really capture that risk across populations. And so before, like I said, is we really perform these models, these JAM models within each population, and then we perform these conditional summary conditional meta-analysis as we went through. We can operationalize that and just sort of incorporate it into a joint model. And really, this is just a mixed effects model where you're modeling now the betas in a second level, call this meta regression. And we're doing it with a JAM framework, and we can incorporate it all in one type of framework here. And we can now do conditional estimates, forward selection, or any selection approach that we want to use within this MJAM framework. The advantage of this is it allows you to use a population-specific sort of reference panel that's appropriate for each summary statistic that you may have from every study. The other advantage here, and I can't go into details, but is this sort of these identity matrices. If you're missing a SNP from a certain population, you can set that to 0, and the approach just sort of naturally incorporates that. And so it'll use the information that it has from any population, but exclude if it doesn't exist. And so now it does that natural, what Elad was getting at is, does it exist or does it not? That takes care of it within any framework we have. And so then the question becomes, now that we have this framework, what do we use as the selection? And one of the things we've been using is this new approach called SuSiE, sum of single effects. And I'm going to just gloss over this, but it's a Bayesian approach. But instead of using this binomial where you sort of flip a coin for every SNP. You flip a coin, and that's what's represented by this multinomial. You flip a coin for a set of SNPs, and you only include that one SNP as you build up the model. And so what's nice about that is it only includes sort of one SNP of highly correlated SNPs, and it allows those highly correlated SNPs to be captured in sort of a credible set. And so I've glossed over a lot of the details here, but that's the general idea. It's computationally fast because they use this iterative Bayesian stepwise selection. So again, it's just like a forward stepwise selection to pick the index SNPs, and then it outputs these credible sets. From simulations that we've been doing with this multi-ethnic SuSiE looks really good. And then we've gone to apply it to real data. And in some of these data sets, we can get some nice figures like this, where we pick out an index SNP. It's a little different than sort of the top SNP from a fixed effects meta-analysis, because again, it's from a meta-regression type of thing. But it does capture that single SNP that can be used in a polygenic risk score, but also the credible sets for that. Now, you'll notice on the y-axis here is the p-values, and those don't seem to be very extreme. What's happening is when we try to apply this to regions where the p-values are 10 to the minus 50, for example, SuSiE breaks down. It doesn't work. And we're still investigating this. We've had conversations with other groups that have looked at within single populations, others, and they're seeing the same thing with SuSiE. And so we've actually modified our approach, and we have sort of another approach to do this that seems to work well in that situation as well. But I don't have, I'll have to come back and give an update on that. But all of these sort of fit within the many flavors of JAM. And we've developed this sort of, they're all hierarchical models of JAM, and we've published that recently here within the, of looking at sort of how you can use the JAM framework to perform sort of Mendelian randomization or GWAS type of an approach. Is you can extend that to a high dimensional omic situation by using HJAM with SuSiE in that framework. And then now what we're doing is using SuSiE with MJAM and doing fine mapping across multiple ethnic groups. I have a few minutes left here, and I'm running out of time. I just want to say that there are a few SNPs that drive a lot of the risk within certain populations. And just to give an idea, this is a SNP within the 8q24 region, and we're looking within individuals of African ancestry. And on the left over here is the frequency of this within the population, within controls. Here, that's the allele frequency in blue. In red is the heterozygote genotype with the variant, and in green is the homozygote frequency of the variant. And if we compare those controls to the cases, you can see that the odds ratio for comparing the heterozygotes is about twofold, but the odds ratio for the homozygote is 5. Okay, and if we then start to stratify the cases in terms of first-degree family history, the odds ratio increases for both the heterozygote and the homozygote. And also for age of diagnosis, if we choose sort of a lower age of diagnosis, the odds ratio increases. So the most sort of restricted case definition over here, familial cases with family history and a young age of onset, we have a fourfold increase for the heterozygote genotype, and over 30 odds ratio for the comparison of the homozygote. So this particular SNP has a big impact. I'm going to skip the absolute risk curve here, but what does that mean is in terms of the familial relative risk, if we think about, well, how does it contribute to the overall impact of this? If we look at the 180 known variants at the time we did this analysis, we could explain 28.5% of the familial relative risk in African ancestry men with all 180. With this one SNP, it explains 9% of that familial relative risk. So that's a 32% of the amount that we can explain is driven by this one SNP in African ancestry individuals. And that impact is substantial on that overall estimate within the 90 to 100% compared to the sort of the 40 to 60%. The gray bar here is when you include that SNP in the genetic risk score. The red here is when you exclude it. And there's a substantial drop in that odds ratio, and it's based on this one SNP, and it's a function of the frequency of that SNP and then also the weight that's captured with that SNP. I'm going to skip forward here so when we've looked at the role of rare genetic variation, we've identified some variants here for within sequencing data that we have. And then we've looked at those genes, and we perform a sort of a burden test within HOXB, BRCA2, ATM, and CHEK2. And we've done that sort of within a discovery data set that we have of a few thousand cases and controls. And then we've gone into the UK Biobank to look at sort of what's the combined impact of the polygenic risk score with rare variation. And the dark line here on the bottom are those individuals that are non-carriers of that of any variants within these genes, and sort of what's the frequency within each one of those groups, and how the polygenic risk score increases risk here on the odds ratio scale relative to the 40 to 60% non-carriers. Now, the light blue line here is for the carriers. And so if you're in the 40 to 60 percentile and you go from being a non-carrier to a carrier, you have about a threefold increase in risk. If you're in the 90 to 100th percentile group, there relative to this sort of the 40 to 60 non-carrier group, you have an increase in about around 4. We've seen that before. But if you're a carrier in that group, you have sort of an 8-fold increase in risk. And so it's the combined effect of being a carrier within these genes plus then your polygenic risk score that adds to sort of the overall risk. And we can certainly translate this into absolute risk curves and evaluate that as we go forward, of saying, well, if you're a carrier, the dotted line here within the 90 to 80 percentile, now your risk is up within this population up around 60% there. So, and then we've done this again within African ancestry individuals, and our data is not as large, so we can't look at sort of a refined values in terms of the polygenic risk score, but the pattern is the same in terms of sort of the additive effect of both sort of the polygenic risk score, increasing risk relative to sort of the, in this case, the 33 to 67 percentile, and then what's the impact of being a carrier in these genes here, sort of within this sample going forward, and the increase there. And so you can see that it's almost a 20-fold increase for carriers with a high polygenic risk score relative to sort of non-carriers in the intertertile range. So just real quickly here to end this is how do we put this in the context of PSA? And so what we've done is we have the multi-ethnic cohort where we had originally 215,000 individuals. We have blood samples on about 70,000 of those, and we've performed a nested case control for prostate cancer with about 2,000 cases and 2,000 controls, and we have genetic data on some of those individuals. Those across 5 different populations: African American, Native Hawaiian, Japanese, Latino, and white. And then we have pre-diagnostic PSA measured on these individuals. And most of those pre-diagnostic PSA, it's sampled around 4.9 years prior to diagnosis, but there is quite a bit of range within that, about 1 to 18 years. And so we're asking the question of, well, what's the impact of PSA before diagnosis, and how does that relate to sort of incidence prostate cancer? And then what's the combined effect of including polygenic risk score within that. And so if we go forward here and look at sort of the combined effects, so including both a polygenic risk score in a model as well as PSA in a model, how does that capture risk? And again, we're looking at individuals from 47 to 87 years old. And what we're seeing is if we look at all cases, the polygenic risk score for 1 standard deviation increase in the polygenic risk score is a little bit less than 2-fold increase in risk. But the odds ratio for the PSA is over 4-fold, but that's in all cases. And so that's probably reflective of those individuals who have prostate cancer, and then the PSA was sort of undiagnosed, and the PSA within a few years of that diagnosis was elevated due to prostate cancer. So what we did is we excluded cases diagnosed within 2 years or within 5 years or within 10 years. And so the cases in this category over here, they were all diagnosed at 10 years or greater after the PSA measurement. And you can see PSA goes down, it remains sort of informative, but it sort of captures the same amount of risk as the PRS. So in individuals sort of measuring PSA early in life or in midlife, before they get prostate cancer, the PSA is informative, but the PRS is also informative there in terms of the risk estimations. So where are we going with all this? So just a conclusion, sort of we've developed a genetic risk score that I think is effective for populations with diverse ancestries. We're performing the next round of Trans-Ancestry GWAS fine mapping to sort of refine this genetic risk score. We're performing the next round of discovery and for rare variants and to see how that sort of combines to capture risk. We're also looking if we can really capture aggressive prostate cancer with rare variants or common variants. And this is the RESPOND study, where we're now recruiting 10,000 African-American men with prostate cancer, and we're measuring multiple omic data, gene expression, somatic mutations. This is with several individuals that I think are on the calls, Scarlett, Iona, Franklin, and others from UCSF. And then the other thing that we're doing is with John, who's on the call here, we have the Pri-Med Cancer, and this is really a grant to sort of look at how we can develop methods for constructing polygenic risk scores across multiple populations, how we can evaluate them across multiple populations. And one of the things I'm really excited about with this is aim 3 here of how we can estimate absolute and excess relative risk in cohorts with multi-ethnic and multi-ancestry individuals. Because as I said before, those absolute risk curves are great, but they're all pegged to this baseline that is identified and defined by self-identified ethnicity. And what we want to do is sort of identify baseline and absolute risks by ancestry and then construct those curves. And I think we have the opportunity to do that with this sort of grant in the populations and the cohorts that we have that are part of that. So with that, I'll end here. Sorry, I went a little bit long, but certainly I want to thank Chris Hyman, who's the main collaborator for prostate cancer and sort of drives a lot of this. And then the postdocs and the PhD students that we see here, and then within my group and his group, sort of driving a lot of the applied and methodological work. Thank you.